Today, we are going to break down the dihybrid cross. Now, I know it sounds a little intimidating, but it's actually the key to understanding the incredible variety of life you see all around you. So let's get right into it. You know, everything in science really starts with a simple question. After the brilliant geneticist Gregor Mendel figured out how single traits were passed down, he started to wonder, what happens if you look at two things at the same time? Like, say, a pea plant's seed color and its shape. Do those traits stick together when they're passed on, or do they kind of go their own separate ways? To figure that out, we have to basically level up from what's called a monohybrid cross, which just looks at one trait, to something a little more complex. And that right there is our key term for today, the dihybrid cross. It is exactly what it sounds like. Di means two. We're going to follow two different traits at the exact same time to see what happens. So how did Mendel pull this off? Well, he went back to his old friends, the pea plants, and he picked two very specific traits to follow in one big experiment. The two traits he landed on were seed shape and seed color. For shape, the round trait, which we represent with an uppercase R, is dominant. The wrinkle trait, that's a lowercase r, is recessive. Then for color, yellow, that gets an uppercase Y, is dominant over green, which gets a lowercase y. Okay, before we meet the parent plants, let's make sure we're clear on this term, the genotype. That's the actual genetic code an organism has. It's the combination of alleles we write out with these letters. Mendel was really smart about this. He started with what we call true breeding parents. One parent was dominant for both traits. Its genotype was uppercase R, uppercase R, uppercase Y, uppercase Y. And he crossed that with a plant that was recessive for both, lowercase r, lowercase r, lowercase y, lowercase y. And what that genotype actually produces, the thing you can see with your own eyes, is called the phenotype. So, for our parent plants, the phenotype of the first one is round and yellow seeds. The second one, wrinkled and green seeds. Simple enough, right? So, what happens when you cross these two? Well, this first cross is actually super straightforward. The first parent can only pass on an uppercase R and an uppercase Y. The second parent can only pass on a lowercase r and a lowercase y. So 100% of their offspring, what we call the F1 generation, end up with the exact same genotype, uppercase R, lowercase r, uppercase Y, lowercase y. And because those dominant traits hide the recessive ones, they all have the same phenotype, round and yellow. But this is where it gets really interesting. This is the main event. Mendel took two of these identical F1 plants and crossed them with each other. And to figure out what happens next, well, we're going to need a much bigger tool. Welcome to the 16 square showdown. Okay, this right here is the absolute key difference. A plant that's heterozygous for one trait, like uppercase R, lowercase r, can only make two kinds of gametes. But our dihybrid plant, that uppercase R, lowercase r, uppercase Y, lowercase y, can make four possible combinations. You could get uppercase R, uppercase Y, uppercase R, lowercase y, lowercase r, uppercase y, or lowercase r, lowercase y. Now, there's a really easy trick to remember this. It's called the FOIL method, just like you might have learned in math class. You combine the first alleles, then the outer ones, then the inner ones, and finally the last ones. And bam, that gives you all four possible gamete combinations every single time. And those four combinations are exactly why we need this big 4x4 Punnett square. We put the four possible gametes from one parent across the top and the four from the other parent down the side. This sets the stage for all 16 possible outcomes for their babies. So once you fill in that whole grid and start counting up what the offspring actually look like, their phenotypes, this beautiful, predictable pattern emerges. It's a ratio that literally became a cornerstone of genetics. First up, we've got the biggest group. Out of 16 possible outcomes, 9 of them are going to show both dominant traits. They'll be round and yellow. Next, something new appears. 3 out of the 16 are going to be round but green. Now remember, that combination didn't exist in the F1 generation. This is brand new. And we get another new mix. A different 3 out of 16 will be wrinkled but yellow. Again, a totally new combination of traits that we didn't see at the start. And finally, we have the smallest group. There's just one single box out of all 16 that will result in an offspring showing both recessive traits, wrinkled and green, just like one of the original grandparents. Now, this 9-3 to 3-3-1 ratio isn't magic. It's just probability at work. Think about it. The chance of any offspring being round is 3 out of 4. The chance of it being yellow is also 3 out of 4. So what's the chance of both happening? You just multiply them. 3/4 times 3/4 gives you 9/16. The same exact math works for all the other combinations. It proves the two traits are being inherited completely independently. And that very idea, that these traits behave independently and follow the laws of probability, led Mendel to one of the most important discoveries in all of biology. He called it the Law of Independent Assortment. It's a fancy name for a really simple, powerful idea. Inheriting the gene for seed shape has absolutely zero effect on inheriting the gene for seed color. They are sorted out completely on their own. So, why is this such a big deal? Well, this law is basically nature's recipe for creating variety. It's what allows for brand new combinations of traits to pop up, like those round green peas. This genetic shuffling is the engine of diversity. It's literally the reason why siblings, who come from the exact same parents, can end up looking so different from one another. At the end of the day, the dihybrid cross isn't really just about peas and a garden. It's a perfect model that shows us one of the fundamental ways life works. It reveals how this constant random shuffling of genes creates this incredible vast potential for variation in the world. And that brings us to a final question for you to think about. Just look around you, at your family, at the different kinds of dogs in a park, at the plants in your backyard. How has this simple idea of mixing and matching traits shaped the world that you see every single day?